This study is entitled Polyisoprenylated Cysteine Amide Inhibitors Deplete Singly Polyisoprenylated Monomeric G Proteins in Lung and Breast Cancer Cell Lines. Cancer ranks as the leading cause of death worldwide. It is an important barrier to increasing life expectancy in every country of the world. According to most recent statistics, lung and breast cancer are the most common types, affecting both sexes. Thus, efforts to build a sustainable infrastructure for the dissemination of proven cancer prevention measures are critical. These are the current management options. However, the potency of cancer treatments remains a major problem due to the development of resistance and associated toxicities of these therapies. Rat sarcoma virus or RAS in cancers are mutations in human genes that encode RAS proteins. They are small monomeric G proteins that transmit signal, mediate and activate some of the major pathways, and acts as binary molecular switches. The activation of RAS is important in the regulation of cell cycle as well as transcription. However, a mutated form would lead to permanent active state, leading to continuous cell proliferation, causing formation of tumor. This study focused on these compounds, polyisoprenylated cysteine amide inhibitors or PCAIs. The PCAIs were designed around RAS posttranslational modifications in which a 15-carbon farnesyl tail was incorporated for proper anchoring in the plasma membrane, an amide bioadjuster as a substitute of the endogenous ester to make it more resistant to hydrolysis, and a linker with a terminal ionizable group on the alpha amino to increase aqueous solubility. These are the compounds that we use, the NSL-YHJ-2-27 and the control compound lacking the farnesyl tail, NSL-YHJ-2-62. Western blot assay was done to check or to determine the effect of the PCAIs on the levels of some G proteins, and results show that in the lung cancer cell line A549 cells, a significant decrease in the levels of KRAS, ROA, RAC1, and CDC42 were observed. In the other lung cancer cell line NCIH-1299, KRAS, ROA, RAC1, CDC42 also showed significant decrease in their levels. However, in the breast cancer cell lines, KRAS, HRAS, NRAS, ROA, RAC1, and CDC42 showed statistically significant decrease in the levels of these proteins. Similarly, with the MDA-MB231 breast cancer cell line, KRAS, NRAS, ROA, RAC1, and CDC42 showed a significant decrease in the levels of these proteins. It's very evident that the RAB5A didn't show any significant difference in all cell lines. To determine the effect of the PCAIs on the migration and invasion of cells, wound healing assay was performed and results showed significant decrease in the number of migrated cells in all 4 cell lines, specifically 72, 41, 46, and 68% decrease, respectively. Invasion of cells was also determined by transwell invasion assay, and results showed 40, 44, and 70% decrease in the number of invaded cells. So the potency of the PCAIs to deplete the levels of some G proteins depends on the structure of the endogenous protein. Based on their structure, KRAS and ROA are farnesylated, CDC42 and RAC1 are geranylated, NRAS and HRAS are both farnesylated plus palmitoylated, and lastly, RAB5A is doubly geranylated. So the ease of depletion by the PCAIs treatment will be easiest in KRAS, followed by ROA, CDC42, and RAC1, because they all have a single C-terminal polyisoprenylated cysteine. In contrast with NRAS and HRAS, which have additional palmitoyl groups, this makes them harder to be dislodged, and the hardest would be RAB5A because of the presence of a double geranyl group in its structure. Concurrent with the Western blot results, KRAS, ROA, CDC42, and RAC1 showed depletion of their levels in all cell lines, while the decrease observed in NRAS and HRAS may be due to other factors aside from PCAIs interaction. Lastly, no significant difference was observed in the level of RAB5A, which implicates that the PCAIs can't work on them. So the effects of the PCAIs may be due to physical competitive displacement that may result in more rapid degradation than when they are in complex with other proteins. Therefore, the findings here that the PCAIs can deplete G proteins in cancer cells is a significant alternative approach to dealing with the oncogenic effect of these proteins, as their activities can be simultaneously reduced to normal physiological levels. Thank you.